Hello everyone and welcome to Bioprocess International Spring Digital Week, brought to you by the organizers of the Global Bioprocess International Event and Content Series. My name is Brianna Kosifret and I'll be your host for today's session titled, The Evolution of Effective Contaminant Removal Solutions. First, I'll cover some quick housekeeping items. If you experience difficulties with audio or advancing slides, refresh your screen with F5. If you are experiencing other issues, hit the question mark button to receive assistance. At any time during the presentation, you can submit your questions into the Q&A window on the left-hand side of your screen. In 24 hours, we receive a link to watch the recording of the session. You can also download a few featured articles in the resource list box on the right-hand side of your screen. Now let's begin by introducing our speaker. We have Daniella Steele, PhD. She's a Senior Product Manager for Cell and Gene Therapy for Astrea Bioseparations. Welcome, Daniella. I'll now hand it over to you to begin the presentation.
Thank you for joining me today. My name's Daniella Steele and I'll be discussing the evolution of effective contaminant removal solutions. So I'll be highlighting the features of Astrea Bio's own chromatography resin, PuraBead, demonstrating how the manufacturing process results in a superior product. And I'll give you a few examples of PuraBead resins that address specific contaminant removal. I'll then be discussing how our novel nanofiber-based technology can transform future bioprocessing practices. But first, I'll introduce myself and Astrea Bioseparations. So I'm based in the UK, working as a Senior Product Manager for Astrea Bioseparations, and I've experience working both in academia and in industry in developing cellular products as models for drug discovery. My focus now is bringing novel technologies to market that solve real and pressing issues for the cell and gene therapy manufacturers, and these are specifically around bioseparations for biomanufacturing. So for those of you who aren't familiar with the Astrea Bioseparations story, it was established as a spin-out from Cambridge University in 1987, and over the years it has built up a wealth of expertise around chemical and ligand design as well as adsorbent development for bioprocessing. We have large-scale adsorbent manufacture and can produce batches of up to 1,000 litres in size to current GMP standards. And we offer a range of adsorbents that can provide regulatory support so that you can use these materials in your GMP processes. And as proof of the quality of our products and our customer service, 21 of our bioseparation resins have been incorporated into FDA or EMEA approved manufacturing processes. And today, Astrea is part of the Gamma Biosciences portfolio of operating companies. We have a global team to support our customers' needs, and our state-of-the-art research and development lab is based in Cambridge, UK. We have manufacturing sites based in the Isle of Man, which are part of the British Isles, as well as in Canada. Our US headquarters are located in Boston. Astrea Bioseparations also operates with a number of distributors throughout Europe, Asia, and the Middle East. So now I want to illustrate how Astrea Bio's proprietary chromatography resin PuraBead is helping to address some major concerns in biomanufacturing industry. Firstly, I think it's fair to say that there are very few parts of the biomanufacturing industry that have not been affected by difficulties with supply chain issues, which is why I wanted to highlight that Astrea Bioseparations has been making its own chromatography resin for over 20 years. This means we have very good control of our own raw materials and have systems and processes in place that mitigate supply chain stresses seen today. We manufacture using a continuous process, meaning that the batch size can be determined by the customer's specific needs, even up to 1,000 litres if required. And our manufacturing process incorporates methods and steps dedicated to reducing environmental impact. This enables our customers to reduce the contribution to their waste and pollutants by the industry. To the right is a graph demonstrating the performance of the PuraBead resin compared to a comparable market-leading product. This pressure flow curve demonstrates how the PuraBead resin maintains flow rate at increasing pressure, while the flow rate for the competitor product decreases. This superior flow characteristics are underpinned by a manufacturing process that results in very uniform resin particles. The manufacturing process starts with all non-toxic raw materials of water, agarose and buffer. And unlike the manufacturing of other chromatography resins, the PuraBead process does not require oils or solvents, which are a major waste product that need resources to process. The starting mix is led to the beader that uses spinning disc technology to produce beads of predetermined size. Once the beads are collected, the water is recirculated, meaning that there's a reduced burden on natural resources. The water also passes through a heat exchanger, again reducing the environmental impact. The beads are then hardened by a cross-linking process to create the PuraBead resin with its superior pressure flow characteristics. And here is data to show the size distribution of the PuraBead resin particles compared to that of a market-leading resin. And you can see that the processes used to manufacture the PuraBead create a resin with a really tight size distribution, unlike those of the alternative product. This means that PuraBead not only has great flow characteristics, but often is an easier resin to handle when packing your own columns. A great example of PuraBead resin is Itoxiclear, which is designed to reduce endotoxin burden. So endotoxin is a contaminant issue for many biologics manufacturers. Endotoxin is continuously shed from the outer membrane of viable gram-negative bacteria, and it's also released when the bacterial cells die, and it's ubiquitous in the environment, so it's very hard to avoid. Although the bacteria can be removed with sterilizing grade filters, the endotoxin itself is difficult to remove or inactivate because it's extremely heat and pH stable. High levels are found in bacterial expression systems, and finding unexpected spikes in late-stage batches can be very costly indeed. So it's also an issue for analytics, as the presence of endotoxin can interfere with many assays. It's not a new problem. It's been a known contaminant for many years, but it's becoming more of a concern in recent times due to changes in biomanufacturing processes. The current drive to decrease manufacturing costs by reducing the number of steps in downstream processing may in fact result in fewer opportunities to address the endotoxin burden level. And this is why we have developed an affinity resin using the PuraBead to address this issue of endotoxin. So, Itoxiclear is a cost-effective, non-toxic chromatography resin that is scalable from lab scale to large manufacturing sizes. And one key feature of this resin is its large capacity for capturing endotoxin, more than 1 million endotoxin units per mil. So Itoxiclear is ideal for processes involving feed streams from bacterial cell expression, where endotoxin burden can be particularly high. But as endotoxin is ubiquitous in the environment, it is also relevant for research and process optimization purposes to avoid those spurious effects of endotoxin on analytical assays. Itoxiclear also typically has very low affinity for target protein. This means that if you come across endotoxin in a late stage batch, you can reduce the endotoxin burden without the loss of your product. Itoxiclear performs well under a range of pH conditions and is a very useful tool to clear endotoxin from pH-sensitive proteins. So Itoxiclear can play a very useful part in many stages of biomanufacturing, either as an intermediate step or as a polishing step for the removal of endotoxin itself. This table demonstrates a little bit better how Itoxiclear performs compared to other methods such as ion exchange or affinity endotoxin removal membranes. The ion exchange membranes in this study reduced the endotoxin burden to less than 0.06 endotoxin units per mg protein, but it had poor performance due to low protein recovery. In contrast, the affinity endotoxin removal membrane had a high protein recovery, but its endotoxin removal was poor. Itoxiclear, however, was able to reduce the endotoxin burden to less than 0.06 EU mg per ml protein, together with a high protein recovery. And so it effectively had high protein recovery and high endotoxin removal. This product is available in 5 and 50 ml pre-packed columns as a cost-effective and readily available solution for endotoxin removal. So you could have this on the shelf for when or if you experience an endotoxin burden issue. Another useful tool to remove biomanufacturing contaminants is hydrophobic interaction chromatography, or HIC. This is a very versatile purification technique, and it's used across many targets in the biopharma industry, such as with the polishing of monoclonal antibodies. The specificity of an HIC resin for a particular target can be modified through the chemical characteristics of the HIC ligand and the salt conditions that are used. Therefore, a typical strategy in process development would be to screen a number of HIC ligands representing a range of hydrophobic characteristics, as well as a variety of salt conditions for the HIC purification step. Astrea Bio has a range of HIC resins, including butyl, hexyl, octyl, and phenyl ligands. And we've recently launched an HIC screening pack containing each of the 4 resins in a pre-packed column to enable end users to screen their process. This allows you to select the ligand and the conditions that best suit your target protein. And these are available in 1 and 5 ml sizes. And here's an example of such a screen where the aim was to develop a polishing step to remove dimers from a monoclonal antibody feedstock. The initial screen involved collecting fractions of a monoclonal antibody feed that had been run on HIC columns with butyl, hexyl, octyl, and phenyl ligands. In this run, the monoclonal antibodies were removed with a gradient elution step. And as you can see from the gels, the phenyl ligand was identified as being able to remove the dimers. This process was then further optimized using the phenyl ligand resin and a step elution. From the chart, you can see that the phenyl HIC resin was able to reduce dimers in the eluted monoclonal antibody pool to less than 1%. So now I would like to move on to how Astrea Bioseparations is addressing the needs of newer targets that are emerging, specifically in the areas of cell and gene therapy. As we all know, the interest in developing cell and gene therapies is growing very rapidly. And for many companies, drivers such as being first to market are especially important. So manufacturing speed is a significant concern. Many biomanufacturing processes for these new modalities, however, are reliant on legacy products developed for antibody bioprocessing. There are many components of a manufacturing process, but downstream processing in particular is affected by using these legacy tools. This results quite often in long processing times with low yields due to low binding capacities and high associated costs. At Astrea Bio, we've been asking, does it have to be this way? And what can we do to improve this? We can start by understanding some of the causes for these issues, of why legacy bioseparation tools are not ideal. One outstanding characteristic of the vectors used for cell and gene therapy is their size. The size of a traditional antibody is very small compared to that of modalities such as AAV, lentivirus, and exosomes. This illustration to the right shows how chromatography resins have a large surface area for binding target proteins, and much of this is situated within the pores of the resin. Target molecules are required to fuse through the film and pores to access ligands for binding, and this works well for small molecules. The large size of cell and gene therapy vectors, however, restricts their access to the resin pores, and so much of the binding potential is effectively lost. This then leads to longer processing times, impacting the quality of the vectors, and also presents issues with low resolution. Furthermore, process developers are using oversized columns to overcome these shortfalls. And it's this mismatch of substrate format and the target vector size that can therefore lead to the long processing times, low binding capacity, and high associated costs mentioned earlier. So clearly there is an unmet need for purification solutions specifically designed for cell and gene therapy modalities and bioprocesses. And to improve on legacy technology, we have been working very closely with Nanoparel, a Gamma Sciences operating company, who have developed an Electrospun nanoscale membrane suitable for chromatography. Together, we are developing commercial products specifically designed for the cell and gene therapy modalities using Astrea Bioseparations ligand design and adsorbent development expertise. So this image to the right is an electron micrograph of the Electrospun nanofibers. As you can see, the nanofibers provide a very high surface area, so allowing for high binding capacity. The surface area for binding is also instantly accessible, as no diffusion is required, and this negates the need for long residence times. You can also see that the nanostructures are highly porous, permitting even large modalities such as lentivirus to access the binding surfaces. This also allows for very high flow rates. This is particularly advantageous for the downstream processing of large volumes of feedstocks with the low titers that are commonly seen in cell and gene therapy manufacturing. As I mentioned, we are development stage right now, but I'd like to show you some of our preliminary data to help demonstrate how significant an advantage these nanofibers can bring to the bioprocessing field. Here is an example of a study to capture BSA using a commercially available Q membrane adsorber and the nanofiber membrane functionalized with a Q chemistry. Membranes were loaded with 1 mg per ml of BSA to 10% breakthrough, and you can see that the nanofiber membrane captured up to 300 grams per litre at a second residence time, showing the vastly increased binding capacity and even which was even high at high flow rates. This is a significant advantage in bioprocessing capabilities compared to the commercially available Q membrane adsorber, which captured only around 100 mg per ml and needed a residence time of more than 10 minutes. So using the functionalized nanofiber membrane would allow the end user to benefit from very high productivity gains with substantial time and cost savings. Here is the result from a study to demonstrate the stability of the nanofiber membrane. Over time, chromatography adsorbents such as resins or membranes may become contaminated by a variety of protein and non-protein species during a purification campaign. This contamination can affect the performance of the chromatography adsorbent and lead to issues with back pressures, poor resolutions, and loss of yield. The use of sodium hydroxide is a common cleaning strategy to maintain the performance, but this can also have a detrimental effect on the adsorbent performance if it's not caustic stable. This study therefore looked at the binding capacity for BSA and the pressure of the nanofiber membrane after extended exposure to sodium hydroxide, and performance was measured at multiple points over a 24-hour period. And as you can see, both the binding capacity and the pressure were consistent over this period, showing that this product is caustic stable. I think also it's worth noting the significantly fast operational speed achieved. The flow rates were 4 mL per minute, that's 40 membrane volumes per minute, and is equivalent to a residence time of 1.2 seconds, which is magnitudes faster than the times required for a traditional resin or membrane adsorber. So in the previous slides, I've discussed how this new nanofiber technology is presenting opportunities for bioprocessing at speed. But how does this affect modalities such as lentiviral particles, which have a sensitive membrane that can be affected by shear forces? So here I wanted to demonstrate that the nanofiber membrane structure is compatible for processing large membrane vectors such as lentiviral particles. In this study, we used a nanofiber membrane functionalized with a weak anion exchanger that was housed in a spin column device. This work was done in collaboration with the biomolecular core at Edinburgh Bioquarter at Edinburgh University. The purification was performed at lab scale with benchtop centrifuge using clarified lentiviral feedstocks. The samples were then either concentrated directly or included a step to purify the lentiviral particles using the nanofiber spin columns. To demonstrate the functionality of the lentiviral particles, these were used to transduce HT1080 cells with a GFP genetic payload. The HT1080 cells were chosen as these can be more challenging to transduce and so would give an opportunity to highlight any decrease in lentiviral functionality. The microscope images to the left show a field of view for each of the conditions. The first images to the left show the actual cells. The middle images are the GFP signal in green, and then the images to the right are the overlay of the two images, which illustrate which cells had taken up the genetic payload. And as you can see, the performance of the lentiviral particles that were purified by the nanofiber membrane were not compromised at all by this technology. So taken together, these preliminary results indicate that nanofiber technology can bring substantial improvements in bioprocessing capacity and speed. And this novel membrane is highly suited for the purification of cell and gene therapy vectors. This will enable the industry to vastly improve their downstream processing and ultimately drive down costs for patients. So to summarize, Astrea Bioseparations manufactures a wide range of bioprocessing tools, providing security of supply in these times of supply issues. And we provide bioprocessing tools for target protein capture as well as contaminant removal. And finally, we are developing a number of products specifically designed to address the bioprocessing needs of cell and gene therapy vector manufacturing. These are based on novel nanofiber technology with capabilities to significantly decrease bioprocessing times and reduce costs. And with that, I'd like to finish my presentation and thank you very much for your time today.
Thank you, Daniella, for an excellent presentation. We've received a few questions already, but we'll give the rest of you a moment to enter your questions into the Q&A box on the left-hand side of your screen. Before we begin today's Q&A, I'll run through some brief announcements. First, I'd like to thank our sponsors for sponsoring this digital week, including Astrea Bioseparations. And next, I'd like to draw your attention to the resource list box where you can download a featured article, and you can also find the entire Bioprocess International schedule for the remainder of the year. Now let's go back to Daniella for the Q&A. Our first question here, can you explain more about the pH range that Itoxiclear can be used and how often it can be reused?
Thank you for your question. The Itoxiclear adsorbent binds irrespective of pH, so it can be used to reduce endotoxin burden in feed streams that have, pH-sensitive target proteins. But it shows optimal binding between pH 4 to 8. It can bind endotoxin outside of this range, but obviously your target protein may also start to bind. And the second part of that question, yes, Itoxiclear can be reused. It can be used multiple times. We recommend sanitization, however, with 0.5 to 1 molar sodium hydroxide.
Great, thank you, Daniella. Our next question here, you say that the nanofiber membrane is different than other membrane absorbers. What is the reason behind this difference?
The reason why the nanofiber membrane is so different relates to the manufacturing process. It's an Electrospun material, and the process allows us to have control over the porosity of the material. And it's this porous characteristic that really differentiates this technology.
Perfect, thank you. Another question here in the queue. Are there products already available that use this nanofiber technology?
Yes, we're in the late stages of product development at the moment, and we'll be releasing more information about a product launch later in the year. However, if you are interested in our early access program, you're very welcome to contact us via the website.
Great, thank you, Daniella. That will wrap it up for us today. If anyone submitted a question that wasn't addressed, keep in mind that the speaker will reach out to you directly. This session was recorded. You receive a notification in 24 hours when the on-demand session is available for viewing. Please take a moment to complete the feedback form so we can continue to improve your Digital Week experience. On behalf of Informa Connect Life Sciences,